Namaskar. Today we'll study nucleic acid synthesis. So nucleic acid is of two types, deoxyribonucleic acid and ribonucleic acid. So DNA and RNA. Again, RNA is of three types, messenger RNA, transfer RNA and ribosomal RNA. So here the basic units are sugar, base and phosphate. So this nucleic acid is a polymer of your nucleotides, and this nucleotide consists of one sugar, one base and three phosphates. Nucleotide, one sugar, one base and three phosphates, and during the bond formation there will be breakage of two phosphates. So one phosphate will remain and that will form this bond. So we will have sugar, phosphate, sugar, phosphate, sugar, phosphate like that, and to each sugar, in DNA it is deoxyribose sugar and in RNA it is ribose sugar. To each sugar one base is attached, and the bases are adenine, thymine, cytosine, guanine in case of DNA. In RNA the thymine is replaced by uracil, so the bases in RNA are adenine, uracil, cytosine and guanine. And DNA is a double helix structure with two strands, whereas RNA is a single strand. And in DNA what happens, the base adenine is forming bond with thymine, a double bond, the hydrogen bond, double bond is there. Then the cytosine forms triple bond with guanine. So ATGC, and between AT there is double bond, and between G and C there is triple bond, and these bonds are hydrogen bonds. So this is the difference between your DNA and RNA. So the first difference is in sugar. In DNA it is deoxyribose sugar, in RNA it is ribose sugar. Second difference is base, ATCG in case of DNA, and T is replaced by U in case of RNA. DNA is a double helix structure, two strands are there, and RNA a single strand is there. So here you can see one strand of RNA, bases are A, U, C, G, and here you can see bases like A, T, C, G, and double bonds A and T, double bonds are there, G and C, triple bonds are there. So first this DNA, if we see this DNA is having two strands, one is 5' to 3' and the other is 3' to 5'. So one strand acts as the sense strand and the other strand acts as the antisense strand. So this antisense strand acts as the template from which the messenger RNA is synthesized by the action of DNA dependent RNA polymerase. So one strand undergoes transcription and the other strand does not undergo transcription, and you can see this mRNA formed will have your complementary bases T, A, and A for U. So we know A forms bond with T. So in RNA we don't have T, we have U. So we will have U. Then C for G, C for G, C for G, like that, G for C, A for U, T for A, like that, this mRNA. And if we compare now this mRNA with the DNA, the sense strand will have just same base sequence, this mRNA and sense strand will have same base sequence. Only difference is T will be there in the sense strand and U will be there in the messenger RNA. So the enzyme is DNA dependent RNA polymerase. So the antisense strand acts as the template on which this RNA or messenger RNA is synthesized, and if we see this example, the mRNA will have same base sequence like that of the sense strand, because sense strand is the complementary strand to this antisense strand, but the difference is this T in sense strand is replaced by U in the mRNA. So if the enzyme is DNA dependent RNA polymerase, then there will be RNA synthesis. You can see here the base after first thing the DNA double strand will be converted to a single strand. So that is done by the on-winding of the double strand DNA. So it will produce two different strands, and the enzyme involved here is called DNA gyrase or topoisomerase. DNA gyrase or topoisomerase, which separates the two strands of DNA. Now, out of the two strands, one strand will be the sense strand and the other strand will be the antisense strand, and the antisense strand will act as the template. Suppose this is the antisense strand, then it will act as the template, and you can see here if A is there, then here in RNA you will have U, C, in RNA you will have G in DNA, so you will have C, and T in DNA, so we have A in the RNA. So in this way RNA will be produced. But in DNA synthesis, both these strands will produce complementary strands, and from one DNA, two DNA will be produced. So from that we can produce two DNA, and the enzyme here will be DNA polymerase. So if the enzyme is DNA polymerase, suppose here A is there, so A will bind with T, a double bond will be formed. So the nucleotide with T will come, and two phosphates will be released, and this bond will be formed. Then next we have C, then it will form triple bond with G. So the nucleotide with G will come, and there will be formation of this like this. Again there will be release of two phosphates. So in this way this DNA polymerizes each strand, and so from one DNA we can have two DNA. So this is all about nucleic acid synthesis. Thank you.